Hello everyone, this is part 1 of Unit 7 UGC NET JRF Forensic Science. In this part, we will discuss about hair and fibers. Hair is a fibrous outgrowth from the skin of the mammals and it is composed of 65 to 95% protein, up to 32% water, 1 to 9% lipids, and less than 1% pigment and trace elements. The study of hair is known as trichology. The lipids in hair are principally from sebum and consist of free fatty acids and neutral fat. Now, why hair is considered as a good evidence or as a good subject? Because of its resistance to chemical decomposition. It retains structural features as it decomposes. It is very difficult to decompose the hair evidence. With follicle, hair can provide DNA fingerprinting and it is considered as an individual evidence, but without follicle, hair is considered as a class evidence and it gives idea about the mitochondrial DNA. Hair consists of two parts, shaft that is above the skin and follicle that is beneath the skin. Papilla is present at the end of the follicle to provide nutrients for the growth of hair and it is surrounded by the bulb. Sebaceous glands secrete oil, erector muscles stand hair upright and they are present in association with the bulb. Hair shaft is composed of keratin protein and it consists of three layers: cuticle, cortex and medulla. So these are the three layers of hair: cuticle, cortex and medulla. Human hair has cuticle scales which are flattened and narrow. So these cuticle scales are known as imbricate type. Structure of hair: this is the hair shaft and this is the follicle. This is epidermis, the root of the hair, erector muscle, sebaceous gland. This is the dermis part and then the subcutaneous part. Hair follicle wall, hair follicle, the bulb, papilla attached to the arteries and veins to provide nutrients, dermal blood vessel, fat that is present in the subcutaneous layer. Starting with the outermost layer of hair, that is cuticle, its main function is to maintain hair's structural integrity, binding, and prevents the transfer of soluble substances. This layer consists of scales. The scale will start from this end attached to the skin to the other end. So, when we have hair as an evidence, these scales would help us to determine whether or what is the direction of the hair. So this direction of scale point shows the younger and older ends of the hair. The point near the skin would be younger end, from distance to the skin would be the older end. The main types of scales found in cuticle are coronal or crown-like, spinous or petal-like, and imbricate or wave-like. Imbricate type is observed in human hair. The next layer is the cortex. So cortical cells consist of spindle-shaped fibrous structures called microfilaments or macrofilaments, nuclear remnants, cortical fusi, and pigment granules. Macrofibrils are made up of protofibrils that consist of low sulfur proteins containing short sections of alpha helical proteins in a coiled manner. Nuclear remnants are small, elongated cavities near the center of the cells. Cortical fusee is the small air space or vacuoles. The pigment present is melanin and it is of two types: eumelanin that produces brown to black color, pheomelanin that produces yellow to red color. So when you bleach your hair, these pigment granules are removed. Cortex is responsible for the specific elasticity and tensile strength of the hair. In human, the cortex is two-thirds of hair's width. Medulla is composed of shrunken cells which may or may not contain pigment granules. Major amino acids present in keratin are cysteine, serine, glutamic acid, threonine, glycine, and arginine. Proline is a proteinogenic amino acid. It is a secondary amine and is present in hair medullary protein. The various medulla patterns are continuous, one unbroken line of color like this, interrupted, with regular intervals, interruptions with regular intervals, fragmented or segmented, irregular intervals, solid, pigment area filling with both cortex and medulla, none, no separate pigmentation in medulla. Types of bonding. First is hydrogen bonding. Hydrogen bonds are easily broken with water and heat. So these are responsible for overall shape of the hair and also for 30% of its strength and 50% of its elasticity. As you have observed that when we wash our hair, so when they are wet, they are straight, and during temporary straightening with heat. So it is the temporary conversion. Others are salt bonds. These are present in abundance in cortex. Salt bonds are formed when one amino acid links with other amino acid from the positive end of first and negative end of the second. They are responsible for one-third of hair strength and can be broken by change in the pH in both acidic as well as alkaline direction. So these are easily broken when uh, a substance of pH 5.5 or higher is applied. Disulfide bridges or disulfide bonds are the strongest, and the protein structure of the hair shaft are held together by chemical bonds known as disulfide and hydrogen bonds. The more the disulfide, the curlier would be the hair. These bonds cannot be broken by water or heat. Only chemical agents are able to break them. The various types of human hair. Primordial hair are of the fetus. They begin by the third month of gestation, grows on upper lip, eyebrows, palm and soles of fetus, and develops at 9 to 22 weeks. Lanugo hair are fine, soft, unpigmented, non-medulated hair of newborn that are found usually up to 6 months of gestation and shed by 36 to 40 weeks. Vellus hair are fine, short, unmedulated hair which cover the entire body. They do not have any sebaceous gland. Terminal hair are same in both can be same in both sexes, that is scalp hair, hair under hormonal influence and behave as an ambosexual character, that is to determine the sex. Example, pubic or axillary hair. Hair specific to the sex. Example, beard hair. So the third one would be helpful in determining the sex. The second one would be helpful in determining whether the puberty was there or not, and this is present in both. Now hair from different part of the body. Head hair is circular or elliptical in cross section. Eyebrow and eyelashes are circular with tapering ends. Beard hair thick, triangular, coarse and have double medulla. Body hair oval or triangular. Pubic hair oval or triangular with buckling. Arms and legs blunt tip hair. Hair growth. Growth of hair is by proliferation of cells in the matrix of the follicle. More the cells, more will be its growth, and by the increase in volume as they move into the upper bulb. When hair follicle reach at the top of the skin, the outer layer begins to harden into keratin. The first layer to become firmly keratinized is Henle's layer. The outer sheath, the vitreous membrane and the connective tissue prevent the hair to expand laterally and keeps it upwards so that there is space for more hair to grow. Each hair follicle undergoes recurring cycles of active growth, regression and rest. So this cycle has three phases: growth, regression and rest. Follicular activity is not synchronized, means not every hair is in the same phase of the growth cycle, and this type of cycle is known as mosaic cycle. The life cycle of hair: anagen, catagen, telogen, exogen. Anagen is the period of active growth and it lasts up to 1000 days. The root bulb is in flame shape. Catagen phase stage is the phase of regression. The root is bulb shaped. Telogen stage is the stage of resting where the hair follicle is dominant and hair are easily lost. The root bulb hair is in this stage is club shaped. Late telogen, early anagen or exogen phase is the hair shedding phase. The human scalp contains compound follicles in which two or more hair produced by separate follicle share a common orifice to the outside. So with different origin is from different follicles, but there they'll be they'll come as one from the common orifice. So this is the anagen or active growth phase, which lasts to up to 2 to 6 years, and permanent hair removal can only occur in this stage. These are attached to the root, strongly attached. Catagen or transition phase, uh, dermal papilla will separate from the follicle and it will move towards the upwards. Telogen or resting phase, dermal papilla will be separated from the follicle. Exogen phase, in which new hairs start forming. Dermal papilla move upwards to meet hair follicle again and they begin to form new hair. The percentage of follicles in anagen is 85 to 95%. Its duration is 2 to 6 years. Catagen 1% follicles 2 weeks. Telogen 4 to 14% follicles 4 months is the duration. Now hair disease and their defects. So graying process is related to the tyrosine activity in the lower bulb. Due to the cessation of tyrosine activity, hair start turning gray. Beard hair is the first to turn gray and body hair the last in normal aging process. The other defects are monolatrix, that is beaded hair, pili torti, twisted hair, trichorrhexis nodosa, frayed nodes, trichorrhexis invaginata, bamboo hair, trichondosis, knotted hair, pili annulati, ringed hair. The disappearance of pigments. creates cavities in the shaft, and the eventual compression of these cavities can cause disease in hair shaft diameter with age. Graying of hair can be naturally or due to a disease. It is due to the dilution of melanin pigment. Treated hair bleaching, it removes pigment granules and give hair a yellowish color. It make hair brittle and disturbs the scale on the cuticle. Dyeing changes the color of the shaft. The cuticle and cortex both take the color of the dye. It is not because of any other pigment. Poison, it is possible to estimate when the hair was last treated if the entire hair is recovered in an investigation, as human hair grows at the rate of about 1.3 cm per month or 0.44 millimeter per day. Yellow hair are due to heavy smoking, greenish cast due to copper exposure, blue hair, cobalt exposure, blue tint, indigo exposure. In case of slow poison or in case of repeated exposure to the poison, there will be trace elements of the poison found in the hair. Ethnic groups and their hair characteristics. So in Caucasoid or Europeans, the hair appearance is generally straight or wavy. Pigment granules are small and evenly distributed. Cross section is oval or round of moderate diameter with minimal variation. Color may be blonde, red, brown or black. In Mongoloid or Asians, the appearance is straight. Pigment is densely distributed. Cross section is round with large diameter. Shaft tends to be coarse and straight, thick cuticle and continuous medulla. Negroid or African hair is kinky, curly or coiled. Pigment granules are densely distributed, clumped, may differ in size and shape. Cross section is flattened with moderate to small diameter and considerable variation. Human hair versus animal hair. In human hair, pigmentation would be denser towards the cuticle. In animal hair, the denser pigmentation would be towards the medulla. Pigments. In human hair, one color along the length. In animal hair, there are ovoid bodies. The color is present in patches or solid masses. The banded pattern of color is observed. The medullary index, or the ratio of diameter of medulla to the entire hair, it is also known as medullary diameter. So it is less than 0.5 in case of human hair and more than 0.5 in case of animal hair, as the medulla occupies 1/3 of human hair space and about half of the animal hair space. The forking tip of the bristle hair is indicative of Sudae, species of pigs and hogs. Identification and comparison of hair samples using microscopy, comparison, phase contrast, fluorescence and SEM, that is scanning electron microscope. Casting of hair is done by embedding the hair in a soft medium, example clear nail polish or softened vinyl. When the medium has hardened, the hair is removed and it will leave the pattern on that medium. This casting is used for the examination with compound microscope. Neutron activation analysis identify element of hair in case of poison or other things, etc. Moving on to fibers. So these are the fundamental unit of a textile or garment and can be used as a trace evidence, like fiber present on weapon may help in establishing the link of weapon with the crime and criminal. Fiber traced on a vehicle may establish link in hit and run cases. It can also tell us about The occupation of the person whom it belongs to. In assault or rape cases, these may help to identify the assailant. Cordage in the case of fibers and strings may be encountered in case of hanging, strangulation, kidnapping cases. Rope is usually made up of hemp, jute or coir. So these are the advantages of fiber in forensic examination. Fiber classification. They are divided into two, natural or man-made based on their origin. Natural ones are further divided into cellulosic or plant fibers, animal fiber, mineral fiber, and natural rubber. Cellulose or plant fiber can be seed hair, example cotton, bast hair, example flax, leaf fiber, example pineapple, nut husk, example coir. Animal fiber can be animal hair or animal secretion. Animal hair, example wool. Animal secretion, example silk. Mineral fiber, example asbestos. Natural rubber, example rubber. Man-made fibers are further divided into four. Man-made cellulosic, example rayon. Man-made modified cellulosic, acetate. Man-made non-cellulosic, nylon and polyester. Man-made mineral, example glass. Based on their formation, the various type of fibers are natural, which can be animal or vegetable. Example of animal natural fibers are wool, Kashmiri wool from Pashmina goat, silk, mohair from Angora goat and other animal hair. Vegetable, cotton, linen, hemp, sisal. A vegetable or you can say plant fiber. So sisal is agave or leaf fiber, jute, bast fiber. Regenerated fibers are rayon, made up of cellulose. Synthetic fibers are nylon, dacron, acrylon, etc. Miscellaneous fibers are fiberglass, asbestos, etc. Aramid fiber is made from aromatic polyamide and it is a heat resistant fiber. Fibers and their melting point. So acetate, the melting point of acetate is 245 to 260 degrees Celsius. For vinyl, it is 200 to 260. For nylon, it is 210 to 230. For triacetate, it is 288 to 300. Rayon do not melt. Fibers and their scientific name: jute, that is stem fiber, Corchorus capsularis; hemp, bast fiber, Cannabis sativa; henequen, Yucatan sisal, Cuban sisal, it is a leaf fiber; agave, Phorcroides; sisal or leaf fiber, again agave sissalana; coir or seed fiber, Coco nucifera; abaca or leaf fiber, Musa textilis; kennef or bast fiber, Hibiscus cannabinus; cotton, Gossypium hirsutum; linen or stem fiber, Linum usitaceum; pashmina, Capra hircus; shahtosh or Tibetan antelope, it is Pantholops hawksoni; Angora rabbit, Ori Oryctolagus cuniculus. The characteristic features of various natural fibers. So cotton resembles a twisted ribbon when it is observed under the microscope. Mercerized cotton is cotton treated with a strong caustic solution or sodium hydroxide that produces swelling to the fiber, reduces neps, produces twist, increased luster, and the sheen is greatly increased. So it will give the silky appearance to it and it is more fluffy. The twisted ribbon effect almost disappear when the cotton fiber is chemically treated. Ballooning with cupra ammonium solution is useful in the identification of cotton, in which light blue balloons are observed. The reagent is 2 gram cupric hydroxide, ruthenium red, and 25% 100 ml ammonium hydroxide. Linen or flax fiber, these are the vegetable fiber or plant fiber. and they do not have scales. Their characteristic feature is the appearance of nodes which resembles bamboo, and these are also known as bamboo fibers. Coir is a versatile natural fiber. It is extracted from mesocarp's tissue or husk of the coconut. Generally, coir fiber is of golden color, hence it is known as the golden fiber. So it is the outermost covering of the coconut. Mineral fibers are used for insulation purposes, example asbestos and glass. Asbestos is made up of thin microscopic fibrils of silicate minerals. Nylon is the first synthetic fiber and its monomer are adipic acid and hexamethylene diamine. Cotton, linseed and safflower seeds are used for paint oils. Scales are found on wool. The various types of rayon fiber are: first, cuprammonium; second, polynosic; third, viscous. Polynosic is the high wet modulus rayon and a modified version of viscose, that is, it is stronger when wet and it has the ability to be mercerized like cotton. Viscous is made up of rayon plus natural resources like wood or other agricultural products. Rayon, acrylic and aramid fiber do not melt. The various factors for fiber examination are physical fit, dimension, shape, color, nature of material, pattern of weave, pattern of dyeing, thread count, direction of twist, thread used for sewing, weaving defects, etc. Fiber examination can be done using simple or compound microscope, scanning electron microscope, pyrolysis MS, thermal analysis, etc. The effect of burning on various types of fiber. So wool shows singeing, cotton catches the fire, nylon melts, and asbestos has no effect of burning. Thank you for watching. Stay tuned for more such videos. Like, share and subscribe my channel Vidhi Vigyan. Thank you.